Oh, very, very nice. Thank you very much. Okay, awesome. So let's just jump right back in. I'm going to share my screen on the iPad. And just for those watching the recording, just to get everyone filled in, what we're going to be doing is I'll be going through the part that Dr. Larea was able to, that we were able to hear in the recording, the audio recording. We did miss out on the other half of it, so I'll be reaching out to him just to get a better idea of exactly how deeply he wants us to go into that second half, because I'm not really exactly, he's very good at explaining um each slide in depth, like using like few slides to say a lot. He's an awesome instructor, so, but that makes it a little tough to know exactly where he's going in the second half. So we're going to go through what we can today. It'll be a relatively shorter session. In the meantime, do reach out with specific questions about anything he discussed. And in that separate video I make, which will be going step by step through these pathways and blood coag, I'm going to try to be as thorough as possible so there aren't too many questions. But as always, just reach out either way. I'm always happy to help in that way. Okay, so let's just jump in. So as far as I understand, you guys already did GPCRs, which is the main one, right? G protein coupled receptors are going to be a large part of cell signaling in general. You're going to see that a lot in the clinical aspects as well of your careers in the future. It's a huge part of pharmacology for sure. We have to definitely know all about these cell signaling pathways, and we have to be able to know where to attack them pharmacologically. So that is definitely something important to know. But there are also quite a few other potential pathways that we can use, and one of which, importantly, is the tyrosine kinase pathway. So Dr. Larea, he's one of my favorite lecturers of all time. He's awesome. I have him for histology. And he is excellent in explaining what is going on here, because I will be honest, this is a relatively complicated system that we have going on. Okay, GPCRs are actually relatively simple, in my opinion, at least, compared to the tyrosine kinase, MAPK, all that. It gets to be quite a bit, as you'll see in future diagrams, but there are only certain aspects we're going to ask you guys to focus on here. And I'll be sure to cover that in the recording video that I post either tonight or early tomorrow morning, going to step 1, step 2, step 3, one by one. But now we're just going to talk about tyrosine kinase. Our objectives here, and the things that you're probably going to be tested on, are nomenclature. That's important. And obviously it's important, but that can also be very telling, especially when we're talking about pathways. When we're talking about pathways, nomenclature can, like Dr. Larea said, one word can make an entirely different answer, an entirely different question, right? If you say the word tyrosine in front of the word protein or the term protein kinase, the answer is entirely different. So you need to be familiar with how to distinguish one component in a certain pathway from another component in another pathway, right? And that is especially in sections like this is where things like these little trees come handy, right? So you're going to have, for example, kinases, and I'll go into this as we go along. Kinases, right? And they can break into 3 different kinds, and we'll talk about what exactly they are. And those specific kinds can break up into more specific kinds, and each one of those kinds, right? Each one of those individual types have a pathway of their own. So this would just be a humongous tree if we went through everything. We're only going to focus on a few. But nomenclature, pay attention to that. Pay attention to what's highlighted, because a lot of the time Dr. Larea is great in the fact that he will highlight parts of the objectives, right? These are the objectives. This is what he wants you to focus on. And he'll highlight that on the individual slides to say, look, this is how it ties into the big picture. Remember, big picture is what we want to focus on, especially in these very complicated the minutia of these pretty complex pathways. So nomenclature, important. What else? Explain how signal binding to catalytic receptors leads to activation of signaling pathways and biological responses. Okay, so you have to know the pathways themselves, right? Know the pathways. Oh, he said it right here. Here we go. Canonical pathways. Identify suitable therapeutic targets to block unchecked signaling, right? Signaling that's happening without regulation. That's either constantly on or constantly off because of some type of mutation or defective area in the entire protein. That's important. That's how we're going to solve problems that arise in the human body. If we can identify certain parts that are messed up, we can target them and mitigate those issues, or at least identify them in the first place. So this is going to be important, and he'll point out as well where the therapeutic targets can be. Crosstalk mechanisms among signaling pathways. Crosstalk is super important. Probably something I will save for the recording, the pre-recorded video that I'll post up for you guys. But yeah, absolutely important, right? So let's just move right on into it. Membrane receptor classification. So now we're talking specifically about what are called tyrosine kinases. All right, Dr. Larea, he drew us a little picture here, right? So if we have the membrane, all right, you got your little phospholipid bilayer going on, got your tails over here, amphiphilic in nature in general. So here's your membrane. This tyrosine binding site has an extracellular portion. So here's your cytoplasm. Here's your cell on the inside. You have your extracellular portion, which is where your ligand is going to come in. Yeah. We have our intramembranous, our transmembrane binding site, or our transmembrane domain, excuse me, over here. So there's different domains. When people use the word domain, this actually, I didn't even realize this until like pretty deep into these concepts when I was taking the class. But domain just means a place. When you hear the word domain, like sarcomology domain, the kinase domain, the catalytic domain, it's the place where catalysis happens, the catalytic domain, the place where the ligand binds, right? This is the ligand binding domain over here. Ligand binding domain. This, this trans membrane domain, part in the membrane. And then most importantly, right? So once this as Dr. Larea likes to say, that once this ligand tickles this receptor here, you're going to see a response on this end, on the intracellular side. This is where all the action's happening on the inside. And it's important that you realize this. Okay, so here's the catalytic domain. This is where you're going to have your catalytic activity, right? And this is important because it differs from the GPCR. Remember how the GPCR, if we extend this a little bit, the GPCR had that serpentine with the 7 domains, right? You had one end here. You had one end coming out. These are all individual domains. 1, 2, 3, 4, 5, 6, 7. And they all connect. They all connect. And then you have your outside part. You have your outside part here, your inside part here. G protein binds through the receptor, conformational change, right? Your heterotrimeric, uh, your alpha, beta, gamma subunits are down here somewhere, right? I'm just reviewing just to get you guys a little bit familiar, right? You have your alpha, beta, gamma stay together. Alpha is the one that's going to leave. That's going to do the ATP hydrolysis or the ADP switch, right? We kick off ADP because ATP comes in, and that's what sets off the cascade going down here. Adenylyl cyclase, CAMP, etc., etc. I'm not going to get into it, but notice how this is different. This is a different setup than what we have with tyrosine kinase. Here we have the extracellular domain, the ligand binding domain, the transmembrane domain, and then here's where all the fun happens. This is where all the action happens. After this part is where we're going to see the continuing steps, the following steps that are going to happen. Okay, and I'm going to really just break it down as simply as I can, because this is a complicated aspect of biochemistry, but it is fundamental. Even the simplest organisms must do things like this. So, I'm going to go into this. It might seem like I'm babying you guys, but that's just because sometimes that one word will make all the difference in the world. Okay, now this catalytic activity that I'm talking about is normally inactive, unless what happens? Unless we have the ligand bind, right? If the ligand binds, if that comes in, then normally inactive. Here, I'll put in absence of ligand, right? So inactive in absence of ligand. Once that ligand binds, we're going to see that activity happen. Once we get that receptor ligand complex, that's when things are going to start getting into motion. Yeah. Okay. Now this is where that flow chart thing I was telling you about comes into play. And I'll certainly clean this up in the pre-recorded video, but for now, it's important that we actually do get a general understanding of what we're working with, because we thought that GPCRs were complex, and now we're getting into the part of biochem, okay, and especially when it gets to coagulation, and blood, and especially with these signaling pathways, now comes the heavy memorization. Okay, before it was still a lot of memorization for the first exam, but it was the basics like pH and enzymes and proteins like that. As tough as that is, you can kind of reason your way through. Here, you're gonna have to memorize. You're gonna have to run through pathways. You're gonna have to be able to distinguish which ones have which components. So let's begin with protein kinases. Protein kinases. What does a kinase do? Go ahead and throw them in the chat for your own fun. I might not even recognize it or pick it up, but just, come on. What does a kinase do? You guys know this. Add a phosphate group. You add a phosphate group, yeah. You phosphorylate. Absolutely, yes. So that's important. Phosphorylates. Most of the time, at least for now, we're going to think, okay, it activates. When you phosphorylate a protein or a protein, it'll usually activate it. It'll put it into its active state. And of course, there are other cascades, and sometimes you phosphorylate and it'll inactivate. But for now, let's just think of it in the simplest terms possible. Okay. Some are messenger dependent and some are messenger independent. What does that mean? Okay, well, some of them require a particular substrate, a particular molecule to make them active, right? So we're talking specifically about these protein serine threonine kinases, okay? They're different from the protein tyrosine kinases. I know the title of the slide says tyrosine kinase receptors and protein tyrosine and serine, so threonine kinase receptors. Think of these as similar. Put these into basically the same general group. Okay, there's GPCRs, and then when you talk about these, they're related more so than they are to GPCR. Okay, so let's talk first about these protein serine threonine kinases. We all know. Let's break it down. Protein, it's a protein. Serine and threonine. Okay, these we know are amino acids. By now, you guys know your amino acids. Kinases. Okay, so something is doing kinase action on serine and threonine. What does that mean? Okay, something is going to phosphorylate. Is going to phosphorylate serine or threonine, right? Maybe both. Okay, so that's what those do. In order to make this happen, you either have a messenger-dependent pathway or a messenger-independent pathway. A lot of the time, this is where we come into nomenclature, what we were talking about, where nomenclature matters. The words matter, right? So if you're talking about PKA, protein kinase A, right? We're going to associate that A with AMP, right? If we're thinking PKG, it'll give it away. It'll tell you right there. GMP. Okay, how about PKC? Oh, look, calcium. It's not super easy, but it'll help give things away to you. But you also have to remember diacylglycerol, right? We haven't gotten into lipids yet, but diacylglycerol is there. It's a diglyceride. You know what triglycerides are? So a triglyceride, you have your glycerol backbone, and you have 3 lipid tails. Okay, a diglycerol is when you only have 2. Okay, just to give you guys some perspective of what we're talking about. Okay, IP3. This is not associated with anything in particular, right? IP3 is actually it's really just going to stimulate calcium release, and we'll get into that a little bit deeper when we talk about the PIP pathway and the IP3s and inositol. Again, that'll be discussed. It is mentioned in his slides. I just don't know how deep he went into it, so I'll discuss that in the pre-recorded lecture. All right. And yeah, so we talked about messenger substrates. We talked about the messenger dependent ones. There are also messenger independent, and this is where we're going to spend a lot of focus, guys. This is where it gets this whole part right here, in fact. This part right here is going to be the majority of today's discussion. The RAF, MEK, MAPK, S6 kinase. Right? These are all interrelated. These all actually play a role in tyrosine kinase activity and serine and serine threonine kinases. And we'll see it's a very interweaving, complex pathway, but we'll get through it and you guys will learn it, no problem. Okay, and then next we have separately. So here's one. These are your protein serine. Here's threonine, and then we also have our tyrosine kinases. Okay. With tyrosine kinases, it's a lot easier of a breakdown. You either have receptor kinases or other ones are called soluble kinases. I'm not sure how deeply he went into that either. I do need to follow up with him. And finally, dual specific kinases, where you're going to see both. They're going to have 2 active sites. That's the main thing you have to know. We have 2 active sites here. Okay. And they'll be specific to serine threonine or tyrosine. So what I would like you guys to do, and like in your own way, of course, or assuming it'll help you, break it down. You don't have to look at all these words. Forget about all this business. Break it down. You have kinases. Okay. you break them down into 3 different pieces. Serine threonine. Here, I actually already have it up. Let me go over here. Here. Is that? Boom, right there. I did it for you. Okay. Break it down into things like this, into charts. And then from here, as we learn more, you break them down further, right? We're going to talk more about these. We're going to talk more about these. But for now, this is what we have so far. This is the information we have so far. This is talking, remember, about kinases, phosphorylation. So when you think kinases, think of these 3 main groups and how they break up into these smaller pieces. That's step 1. That's where you have to really just get familiar, okay? Okay, so we phosphorylated. Let's assume we did that. We phosphorylated. We activated. Sure, great. Are we going to stay phosphorylated forever? Do we want that cell signal going on in the cell forever, indefinitely? No, of course not. Of course we don't want it doing that forever. That's terrible. We have to be able to regulate. We have to be able to turn it on and off. So how do we undo this kinase action? What do you use? Phosphatases. A phosphatase. And what's a phosphatase going to do? Remove the phosphate group. Yes. Phosphatase is going to break down. It's going to strip the phosphate off. Nomenclature. Important. Remove phosphate. Okay. So let's just get this into perspective here. We have, do it from here actually. I'll just keep this up, in fact. Our protein, we know that we have a hydroxyl group hanging off. The kinases that we're looking at over here are going to take a phosphate off of the ATP. What does ATP stand for? Adenosine triphosphate, 3 phosphates. There are 3 phosphates linked up together. We're going to only take off 1 P. We're going to take 1 little P off of the ATP. So instead of now, there's not triphosphate. Now it's going to become diphosphate. And again, I'm getting very, I'm simplifying it as much as I can for a reason. You might be like, oh yeah, obviously, but this is important. It's important that we really break it down and fully understand what we're talking about. So the kinase is going to take an ATP, assuming maybe we might have magnesium as a cofactor, and we'll talk about certain phosphatases that require magnesium, for example, or kinases. There's different ones that require different things. They have a metal requirement. Some don't have a metal requirement. So we're going to take 1 ATP. We're going to take that ATP. It's good when it works. And we're going to add it on to the protein over here. This is what a phosphate group looks like. You're going to have the phosphorus in the middle and 4 O's, 4 oxygens. And of course, valence rules and resonance rules dictate that there has to be a couple negative charges and a double bond. We're not going to get into the organic chem of it, but that's what it looks like. Or I think that's just gen chem. I'm not even sure, but yeah. So we lost 1 ATP. Now we have ADP. Another word you should just be familiar with, just on the side, is the word pyrophosphate. Pyrophosphate equals PPI, 2 inorganic phosphates, right? So if I took 2 ATPs off of two phosphates off of an ATP. That would leave me with ATP minus two phosphates would lead me with what? AMP, right? 3 minus 2. Now we have 1, adenosine monophosphate. This is just from the side. This is not really relevant to here, but it will show up many times, so I'm just getting you guys familiar. Plus an AMP plus PPI, and that is your pyrophosphate. Okay, that word will come up, and I can guarantee you it's going to be useful to know that, because some people just assume that you're going to know, and I didn't. I had to look it up. Okay, so we did that. Now we have our protein and our phosphates attached. Now we're going to assume this is on. We're going to assume this now is on. This is activated. Okay. We want to inactivate it. We want to turn it off. We don't want this pathway going anymore. We don't want this protein doing its job and going and doing a bunch of other things over here. So we want to turn it off. What do we introduce? Our phosphatases. And that phosphatase is going to take this here. It's going to take the phosphate that we added and kick it off. It's going to take it off, and naturally, you know, it's just going to rehydroxylate itself back into this protein OH combo. Yep. Yeah, AMP. And yeah, guys, just if you know the answer, or even if you don't know the answer, engage, throw it in the chat. I might not see it, and then, you know, that's okay, you know, but, you know, do it. Just talk, just go. This is for you. You know, I don't need to take this exam, but you guys should be testing yourselves. You guys should be constantly, you know, going for it. Yeah, heck yeah, keep doing what you're doing, guys. Excellent. All right, good. So we get that. You know, I kind of went into there, but we get it. Here's what I was talking about before, right? Sometimes you have metal requirements. These are all different, these phosphatases. So you thought it couldn't break down anymore? Yes, it can. It can break down more. Okay. We thought that these messenger dependent or messenger independent, like we talked about up here, same deal. Messenger dependent for kinases, messenger independent, dependent, independent, used for both phosphatases and for kinases. Same deal. Some of them have metal requirements. Some of them don't. So here they just give them specific names: PP1, PP2A, and they'll tell you the specificity of what exactly they're going to look for, like the beta subunit of the phosphorylase kinase, the alpha subunit. And they, you know, these phosphorylase kinases or phosphatases, as we say, they have different parts to them as well. These proteins have different domains. All right. And so these two, 1 and 2A, they don't need any metals. They can do their own thing. So that means they'd be what? Messenger independent. They're messenger independent. However, if you look at some of the messenger dependent, we're going to see that, yeah, maybe the PP2B, right, or calcineurin, it'll need calcium. PPM will need magnesium. Now, I'm not sure if he wants you to memorize these specific things from the table. I think he might just be trying to illustrate a point that there are many different types, but I'm not 100% sure. So what I'm going to do is star this so I can ask him, does he want you guys to memorize this? And we can address that when it pops up. Yeah. Okay, so now I'll of course let you guys know, and then I'm going to go through all of this again in a much more quick version, just with your quick 1, 2, 3, 4, 5, much neater than all of this. But you guys need something to refer to when you're doing your studying, because, you know, having it all laid out is actually very helpful. Okay, again, the same deal here. Protein tyrosine phosphatases, right? They're going to be either receptor or soluble. Same thing as the kinases. Now we have the phosphatases. They're related. And then also finally the dual specific phosphatases, which means what? 2 domains, 2 active sites. They can do either one of these or both. Okay. So let me ask you guys this. We just finished phosphatase. So if we were to increase the calcium concentration in the cell, what would we see? Based on what you guys just learned, what would we see? If we increase the amount of calcium in the cell, would we see phosphorylation or dephosphorylation occur? You guys are saying dephosphorylation. Okay. Anyone, any other answers? You're half right. Oh, P. Both. Whoa. Yeah. Honestly, you guys all might be right or you might be wrong. Why? It depends, right? Because remember, these kinases also were messenger dependent. Calcium here, the PKC that we talked about. The PKC is calcium dependent. That's a kinase, though. Here, the PP2B, the calcineurin, calcium dependent, but that's a phosphatase. Now, this illustrates a huge point that Dr. Larea really wanted to get across, and importantly so, that there's a time and there's a place for everything, right? So if you're in a certain scenario where you want the kinase active, it'll have to happen at a certain step, at a certain point in the cascade. Because if you just randomly increase calcium, who knows if you're gonna be affecting the phosphatases or the kinases. So the answer is it depends. It can be either one. And it's these little nuances that are going to be the testable portions of the exam that, so that's why it really is important. This might seem dumb to you. This might seem like, oh, okay, yeah, obviously, like you don't have to baby us. We're in grad school, but this is where the little nuances matter, right? This is where the little tiny nuances matter. Knowing that these are messenger dependent, these are independent. Tyrosine has soluble and kinases and receptor kinases. Dual specific has two active sites, one that phosphorylates serine threonine residues, one that phosphorylates tyrosine residues. These little things matter, guys. That IP3 does not have an actual associated messenger. So yeah, it is important that you do delve into the details and you have to understand why they are as they are. And the textbook, guys, is actually incredibly useful as well in this regard. I was reading a little bit of it earlier, and it's really great. Like I don't usually advocate for textbooks too much, but this one rocks. It really does the job. So do take the time to read up on that, and even some of the stuff that wasn't discussed in class, it really helped me out. Like it really will put things into a perspective and word them in a way that, Dr. Larea, he's a master at his craft, and there's no denying that. But sometimes just hearing it or being able to read what was said over, maybe a couple times and go back and refer to a diagram, it really makes a difference. And I'm harping on that for a reason, because these little tools will help. Use the textbook, print out. I think it's page 400 to 460 is chapter 12, which is going to cover all of your receptor stuff, including the stuff that was already discussed, the GPCRs, ion gated channels, which are going to be also just as important. And you'll have it all right in front of you, print it out. If you don't have the actual hardcover book, then you have access online through the library, and definitely print it out. Look at that, use that, because that's what this class is based on. The material in this book, most of these diagrams that you see coming up in the future slides are going to be straight out of this book, and they're going to explain them in words. So use them. Historical perspective, I'm not going to go through that right now. It is interesting stuff. Go back to the video and Dr. Leroy can explain it much better than I'd be able to. Okay, here. Let's see who was paying attention in class. Upon biopsy, neoplastic cancerous neoplastic syndrome is invasion of cells into other compartments, but beside the point. Upon biopsy, neoplastic cells show continued activation of cytoplasmic kinases. Which of the following gene products is likely to be involved in this process? All of them except for P10. I think you said most of them. So let's pretend that we didn't already hear his lecture. From what I just said to you guys just now, what would your answer be? It's a bad question. They'd all be involved, right? They all activate cytoplasmic kinases, right? Even GPCRs. What kinase is involved with GPCRs? Actually, you know what? No, I'm gonna keep those separate. I'm not gonna test you guys on GPCRs. But yeah, all of them. So this is a bad question. Why? Because it can be, any of these can be involved. Here, her, RAF, RAS, P10. These are all mentioned here somewhere. I don't think her is in here. Did he put her on here? No, I don't think he did. Okay, well, yeah, the point stands. All of them, or it could be all of them. So let's get more specific then. What if we said has protein kinase activity instead of is involved in the process? What if we say has protein kinase activity? Oh, good, you guys answered. Yeah, PKA. Yeah, that's for GPCRs. Okay, well, we're gonna find out. All right, it's not going to be all of them. I'll just, I'll give you guys a hint, and then I'll go through the exact answers in the pre-recorded lecture, where I can really delve into the details. But I just want to see how he wants us to go about it, because he did leave that up to you guys. He didn't answer it for a reason, so I want to see if it'll be all right if I answer. But we'll check with Dr. Leroy. He'll know what to do. Again, so what if we even change the word from protein kinase activity to protein tyrosine kinase activity? Oh my God. Okay. That changes it again. Let's go back up. Protein tyrosine. Oh, this is phosphatases. Sorry. Protein tyrosine kinase activity. Oh, okay. So we haven't even gotten into those yet, but it doesn't seem like it's going to be involved with any of these, right? Because those are serine threonine kinases, so we can knock out some of those already off the bat. But the point is, and I'm not going to answer that again. I'm going to see if it's all right with Dr. Larea first. But the point is here, nomenclature, words. That's why it's the first objective. Nomenclature. You guys need to know, because if you just say if you kind of skip over the tyrosine part, right, you're going to get this answer wrong. But if you focus on this particular word, tyrosine kinase activity, you'll get the answer right. Protein kinase activity. They're so similar. They're just so similar. And it's honestly, and then you'll notice in the rest of biochem that the littlest nuances, whether a little tiny phosphate group is there or not, will change the entire meaning of what's happening after that. And that is just the way it is, and that's what honestly makes it so cool, but also so difficult. That's why not everyone's a biochemist, because it will get frustrating at times, but that's all right, because we're all cut out for it. I gotten to know quite a few of you guys, and This is a special group, I can tell for sure. So you guys just keep pushing through. You will learn all of this, I promise. And especially once you finally you finish going back to the beginning, everything will make a little more sense as you go through. Okay, hearing it once from Dr. Larea is not going to help. It's not going to help you guys understand this. Hearing it once from me is certainly not going to help. If Dr. Larea didn't help, you're going to have to do it over and over and over again. All right, point proven. Good. Reversible. Reversible. Keyword. Protein phosphorylation regulates cellular activities. The fact that it can be reversed is important, because if I just phosphorylated something and it stayed phosphorylated, that signal is going to keep on happening forever and ever and ever. Now, what if I eat sugar, right? And I want to turn on the insulin, which is actually an excellent, they use that example in the book, right? So if an insulin receptor binds insulin and everything, and we'll talk about how autophosphorylation, it gets phosphorylated, right? With the kinase. Or what happens when the sugar's gone? I'm going to keep making insulin. I'm going to become hypoglycemic. I'm going to pass out. I'll die. So we got to be able to reverse. Must be able to reverse. That's the key in the regulation here that we're talking about. Regulates. Everything's about regulation, regulation, regulation. When we talk about cholesterol, everything is about regulation. And that's what you need to focus on. Nomenclature helps you understand the regulation, and they all tie into one another. All these objectives. Dr. Larea is he knows what he's doing. And I would recommend that you rewatch the lecture, you know, even with our broken recording for the tyrosine kinase, the second part of cell signaling. It's worth doing the extra work, you know, of playing them side by side, the video recording and the audio recording, and dealing with that extra, you know, hassle, because he really does explain it, and he asks the right questions, which is a key, and he'll help guide you. But maybe the way I explain it will bring a different perspective that can help supplement the way he teaches it. So we're going to go through it. Phosphorylation, it regulates, like it said up here, regulates these things over here. Protein activity. It can either go up or it can go down, like we said before. But more importantly now, what we're curious about here is this part. Subcellular location. And we'll talk a little bit about that. Again, protein kinases are classified as being either protein serine threonine kinases, protein tyrosine kinases. So this might be better. 1, 2, or dual specific, which is basically 1 and 2, right? We talked about in enzymology, because I believe, yeah. So I did enzymes. I taught enzymology with you guys. We talked about the active site. We talked about the lock and key model. We talked about the induced fit theory, right? Granted, this is different, but it's also very similar. You know, now we're talking about cell signaling. Before we're talking about an enzymatic reaction, but this is quite similar, right? You have to have a key that fits the lock. So you're not going to be able to phosphorylate a tyrosine, for example. You're not going to be able to unlock a tyrosine lock using a serine key. And the same goes vice versa. Now, why is that? Bulky. There you go. So you get the idea. Yeah. So look at these shapes here that we drew. Look at these shapes. Oh my God. This only has room for what's shown in here. You only have room for this hydroxyl, this hydrogen here, and this CH2. Nothing else is going to be able to go into that serine. So if you want to phosphorylate that serine, you better be able to fit Same with threonine. A little bit different here now, right? Now we have an extra methane over here, or methyl group. Okay, what about tyrosine? Tyrosine, absolutely. Yeah, it's bulky, it's huge. That's going all the way down here. So that is just kind of to illustrate the point to you guys that binding specificity matters. Now, I'm not sure if it was covered in your first cell signaling lecture, but there's a term called specificity and selectivity, and these terms are important, right? Integration, divergence, I'm not going to get too into it, but amplification, right? These little terms matter. What's the amplification? What is that talking about? Let's tie it in a little bit. Amplification. So, that had to do with our discussion on GPCRs, but it's applicable in general when it comes to cell signaling. You have receptor, ligand, they bind together, right? You have ligand receptor. Ligand receptor activates one thing. That one thing activates three things. Those three things activate 100 things, right? You guys learned about this with GPCRs. Increases signaling downstream. So that one activation of one receptor can end up making 100 or 1,000, if not more, separate actions happen downstream. Amplification. That's why even just turning on one receptor yields a massive difference. Amplification. Selectivity. That's where this comes into play. Words like that we learned about in the beginning, and I'm sure they must have mentioned it in the beginning. Did they say something like that? Do you guys remember the word selectivity being thrown around at any point? I didn't get a chance to actually watch the first lecture. I believe David did cover it because it was on exam one, but it does tie in. You did. Great, great, beautiful. Who can tell me what selectivity means? Just throw it out there. You don't got to get all technical and specific. Just I mean, how about like specificity? Yeah, it needs a specific thing to be activated. It's exactly what it says. It's not a trick question. Specificity is achieved by precise molecular complementarity between the signal and receptor molecules. It has to be precise. And this is what it's talking about. To the molecule, precision. I think I use the word selective, but specific is specificity is a more accurate term. The two are distinct from each other. We'll definitely cover that in the future, and certainly in pharmacology, they're very distinct. For example, if you're curious, the drug has to be selective to the receptor. The receptor has to be specific to the drug. It gets a little bit granular, but that's beside the point. You get the idea. Great. Okay. Timeline. I'm not going to go through this. It is interesting if you want to go through it. His point here is the Sarc domain. The Sarc domain gave rise to many of the subsequent discoveries that made our understanding what it is today. Okay. Here, TD, if it's highlighted, pay attention to it. TD, just know that it means transformation defective. Okay, there's a defect in there. There's some more timeline. I'm not going to go through this right now. Okay. See again, right here. Session objective to be fluent in nomenclature. Nomenclature. The words. What do they mean? How do they apply? Where do they fit into the big picture? This is also important. What are proto-oncogenes versus oncogenes? This is a little bit of a tough one. I'm curious to see, I'm not going to ask too many more questions in the interest of time, but I'm curious to see if you guys can word this into like layman's terms, because we're getting pretty technical definitions under these things. Normal genes in our body that can control different things. What is that talking about? Oncogenes are precursors for oncogenes. Growth factors, cell growth. Well, when you say growth factors and cell growth, what are you talking about? Proto-oncogenes or oncogenes? Give me some specifics. Let's get the brains thinking a little bit. Proto. You guys are good. I tell you, man. I'd be nervous if you guys were in my class. I'll tell you that much. All right. Yeah, you guys are doing. You're pretty on point. Let's look at the actual definition of proto-oncogenes first. We'll leave oncogenes after. Growth factors, very nice, or enzymes that participate in the network of signaling. This is our idea here. This is what we're talking about. The network of signaling and biocellular pathways that control cell division, inhibit cell differentiation, or halt normal cell death, apoptosis. What happens if you halt normal cell death? If these cells aren't dying when they're supposed to, what's happening? Uncontrolled growth, too many cells, tumors, cancer. Absolutely right. You guys are on point. Absolutely right. And that's a big problem. You must have apoptosis. And if you're not able to control it, if you're not able to inhibit cell differentiation, if you don't know which cells are becoming what, that's what differentiation means, right? There's proliferation, which is just taking one cell and making more, right, of that one cell. And then there's differentiation, which is like when we have our stem cells, right? Our stem cells or our progenitor cells, as they're called. And this can become all kinds of different things. And depending on the cell signaling involved, depending on what certain substrates are involved, determine which route it's going to take. Differentiation. So if we're not making the right cells from the progenitors, from those stem cells, we're going to have a whole bunch of heart cells and not enough lung cells, and we're going to be all kinds of screwed up, right? Don't quote me on accuracy there. I'm just making a point. But yeah, cell division, different proliferation, differentiation. We just talked about it. Apoptosis, cell death. These need to be replaced once they get old. Otherwise, they're not going to do the job right, or you're going to have too many. Okay, excellent. Proto-oncogenes. So now let's look at oncogenes. A dysregulated proto-oncogene. Okay, so if a proto-oncogene controls these things, right? If a normal proto-oncogene is responsible for doing these things, these aren't bad. We need these proto-oncogenes. An oncogene, that's when we're talking bad. That is when we have a problem. That's when the good guys turn bad. Dysregulated proto-oncogene. Namely, proto-oncogenes that become overactivated through mutation or gene amplification, which means there's too many copies, right? What is a cancer? Oncology, right? Cancer. Oncogene. Well, a cancer is the uncontrollable growth. The uncontrollable growth of cells, of abnormal cells, I should say. If you want to just get to the most basic definition of what a cancer is. Uncontrollable growth and differentiation and proliferation of cells. Of course, there's a whole cancer biology subject. Oncology is its entirely own thing. I'm making it as simple as humanly possible, but that is what an oncogene is, a messed up proto-oncogene. We all have proto-oncogenes. I hope we don't ever get oncogenes. That's when we have a problem. Too many cells growing. You got it right. Yeah. There you go. Okay. So nomenclature, guys. Important. Pay attention. Dr. Larea is not going to say things that are not important. He's not going to mention anything that does not tie into the big picture. And that's one thing I really admire about him as an instructor, and histology too. He'll ask questions and it'll be a trick question sometimes, right? But I don't feel stupid after trying to answer it. I feel smarter now that I know how to answer it in the future. And so I really encourage you to listen to what he has to say in these lectures, because the way he sets them up is key. It's like, it's awesome. I try to use that in my own classroom, because I teach anatomy physiology down at Marist, and it works. It works. You walk them through it. You walk yourself through it in a way that seems almost stupid, right? You dumb it down, but it really, it's important, and it helps clear things up. It's really great. Oh, there goes Cleo. Cleo does not like oncogenes. All right, puppy, come on. 1 second, guys. Got to do what you got to do. She's about 8 pounds, and she's out here protecting the house. All right. Can we see Cleo? Yeah, if she wanders around this way again and she handles her protective duties, I'll definitely turn on the camera and show you guys. Yeah, she's vicious. No, I'm just kidding. She's a sweetheart. Okay, great. So we did all that. Now we know oncogenes, proto-oncogenes. We know that Sark, right, is going to help give rise to a lot of our understanding in terms of the structural features of tyrosine kinases. Now here, structure and function, and just like anatomy and physiology, structure and function are related, and not just related, they're dependent upon one another. The structure of a tyrosine kinase receptor is, or a non-receptor tyrosine kinase, excuse me, is going to be based on the Sark as like it's the main prototype. That was like the first Iron Man suit that Tony Stark made in the desert, right? Like all the ones that came afterwards that were super cool, they were built off of that one crudely built thing that he made in a cave, right? So that's our Sark. That's what we're talking about here. So let's just go through over here. What was Dr. Laroya talking about? Right. So, and that's what I was saying earlier. When he puts information on the slide, like when I first looked at this, I already took this class. When I first looked at this, I was lost. I had no idea what was being shown here. And then when he explained it, I was like, oh, okay, I understand. So that's why I do want to make the pre-recorded lecture, because some of these slides, he puts them together in his own way. It's not like he just writes out the answers on in words on the slide. He takes a very interactive approach. So that's why I just do want to double check with him. Okay. See, Sark, that's your proto-oncogene. That's normal. We all have a C Sark protein, right? Here's your N terminus, here's your C terminus. That can be turned on and off at the right time, right? This will be turned on and off at when it's supposed to. That's the proto-oncogene, right? What's this over here? What's this green circle? What am I circling? What is that? Tyrosine. Well, what do you know? We're talking about tyrosine kinases. Maybe that has something to do with it. Yeah. Yeah. So why is tyrosine? Don't ask me why, no pun intended, but it's tyrosine. You guys should already know that from your amino acid discussion. And again, you notice how that's tying into now into the first exam information. You need it for the second exam. So yeah, it's not cumulative on paper, but you know what? It is. It is. You have to know what was talked about before. Okay. Domain. Here's that word we talked about. The place, the spot. The kinase domain. What's that mean? Oh, this is the place where the phosphorylation happens. This is the place where we're going to find the kinase activity. Okay, I get it. So this is the 536th amino acid, right? So there are 536 little amino acids leading up all the way over to here. And this section, they didn't specify which section. They do sometimes. This section right here is the kinase domain. That's where these amino acids over here that are lined up, they're going to make up the phosphorylating aspect of the protein. Okay, good. All right, great. So I know what's happening now. Good. All right, so what's this down here? What are we talking about over here? Oh, VSARC. I think the C is for conventional. I'm not 100% sure, but yeah, well, just that's what it is. Then you have viral. Okay, so this is a Sark domain that had some viral activity corrupted, right? It's dysregulated. That's what we were talking about up here. A dysregulated proto-oncogene. So this is a problem. This is a problem. This looks like it could be an oncogene. Why? Oh, it's entirely active. It's always active. Why? Because it lacks regulation. It lacks point of regulation. What's that point of regulation? Tyrosine. Huh. Oh, okay. Tyrosine. That's why they call it that, because the tyrosine determines the regulation. That's why this is important. But wait a minute, it's not even present here. There is no tyrosine here. So if we're lacking the point of regulation, the tyrosine, and that means we can't regulate it, what does that mean? It's always on. It's always active. That is a problem. That is an issue. That cannot be controlled, right? We don't want that. Viral Sark. That viral Sark can turn into a cancer. That's something that we want to avoid. And how can we target it? How do we know? How can we fix this cancer? And yes, we don't know. We don't have the cure for cancer. Why? Because there are multiple cancers and there are multiple causes for multiple cancers, right? But if we know this entire structure and the function as well, you see, structural features that will give us information into the function and, more importantly, into the mitigative action that we can take. How are we going to come up with therapy? What now we know we can target certain parts. What if there's a genius out there who comes up with a way to add that Y, to add that tyrosine over here and fix the problem? Right? That's just what we're trying to understand. That's why we break it down into such minute little pieces, because one of you guys listening right now, you might be the one to figure out how to fix this problem. You might be one of the ones to find out how do we fix this viral form of potential cancer of oncogenes. The key here is not we don't it's not the specifics of this slide. It's the point behind it, the big picture. I can always say that every time, the big picture. All right. We're showing that there are specific little changes and that the tyrosine aspect is the reason it's called tyrosine. That's messed up. There's no regulation. There's no tyrosine. There's no regulation. There's no regulation. There's no way to turn it off. It's a constantly active problem. All right. It's locked in. I'm sure you guys get it by now. Abelson. Okay. A proto-oncogene. Not a problem. Not a problem at all. BCR Abel. Not good. I don't remember what BCR stands for, but BCR Abel's not good. What is not good about it? Why is that? What can you see on that little picture, that diagram, that is different? It shortened the cap. Oh, okay. Okay. So the cap must be important. And we can see here, Dr. Larea explained to us, the cap, and pay attention to these boxes, the cap stabilizes the inactive form. If you're unable to stabilize the inactive form, then you're going to have an active form at all times. Uncontrolled, dysregulated cell differentiation, proliferation, and lack of apoptosis. Or too much apoptosis. Who knows? Depends. Nomenclature. Right? Because it ties into the big picture. No regulation, no point of regulation, problem. That's key here. Let's continue. Talked about all these. Oh, Jack stat pathways are going to become a big talking point as well. I'm not sure if he delved too deeply into those in his lecture, but again, it will be covered. Don't worry. Jack, a proto-oncogene. Normal. It's okay. It's fine. These are regular domains. Except here, we have two. One of them is a kinase domain, catalytic activity. Right? This will do the This will do the phosphorylating. But here we have a pseudo kinase domain. Pseudo fake. Doesn't actually have any catalytic activity. This has no catalytic activity. This one does, though. Okay. And that's just what it is. This is the way it's set up. Pretty big one too. This is 1,142 amino acid residues. Okay, and we can determine which part of which domain we're in based on the numbering. Yeah, you guys get that? So this might be number 642 to 840, and this might be 301 to what's this 40 to about 200 here up to 500, and you have this little tiny space in here. You might not see it, but that little tiny space there is not going to be any either. It's just like a linker portion. Okay, now we have a problem. Jack. Oh, they tell you right here. Here. At the 617th residue, and this is important, you guys can understand what these numbers mean. This matters. At 617, the 617th residue out of 1100, you have a valine, V, being switched out for F. What's F? Phenolphthalein. Or PHE. Yeah, so this is what it's saying. This F is substituting the V. Phenylalanine, substituting valine. Okay. Just it might help you understand, or you just keep track of things in your mind. Janus is the two-faced god, and that is, that makes sense. That's appropriate because there's two domains. I don't know if any of you guys watch The Office. Janus, Michael's boss and then girlfriend and then crazy lady. She's a little bit two-faced, I'd say. So that's just how I remember it. She seems, she's mean and then nice and then mean and then nice and snip snap. I don't know if you guys ever saw the episode, but hopefully somebody watching the recording got a little chuckle out of that. But yeah, Jack. So break it down. Janus and kinase, JAK, Jack. Beautiful, right? Lovely. Yeah, great. Okay. Let's go on. And the coming slides will go a lot quicker, but I do need to break down the importance of these things. So Sark. We said Sark is the prototype. Why is Sark the prototype? It was the first one they found, right? It was just a matter of which one they found. Sark. Okay, we talked about domains. Remember we talked about domains and how they're important? Domains, places. So these different domains, these different places do different things. These different places that do different things require different activators. They require different molecules to make them do what they're supposed to do. Okay, great. So when we say SH1, what are we talking about? We're saying Sark homology 1. The Sark homology 1 domain. Now that means in long, in fancy science terms, the thing that looks like Sark in structure, right? The first domain that we identified that looks like Sark, homology. It looks like it looks like it. It shares similar structural components or makeup to Sark. Okay. Oh, wait, yeah. So here's Sark. Here's Jack. Here's Abel. These are all different proto-oncogenes, right? They all share the same specific domains, right? They all share the same domain. Well, granted, there might be a little bit more space over here on the C terminus, or more space in between the SH1 domain and the next one over, which we'll talk about, right? Sark in particular has a tyrosine portion to it. Now, these are non-receptor tyrosine kinases, right? So, but they're just to illustrate a point to you guys. Okay, so we know what SH1 is. When you think SH1, no one's going to ever say that, first of all. You're never going to hear someone say, oh, the SH1 domain. It's not going to happen. They're going to say the kinase. We rarely say SH1, but it can't be, it has to be technically the SH1 because it's the first part. The kinase is the first part. Okay, what about SH2 and SH3? Oh boy, you're going to hear about these, folks. You're going to need to know what these do. You will definitely need to know what SH2 and SH3 are and how they are different from each other and how they're different from SH1, what they mean. So let's just jump first into SH1 or SH2, excuse me, because SH1 is the kinase, right? Yes, we know that. Beautiful. SH2, Sark homology 2 domain. That is what we're going to use to bind our phosphotyrosine residues. Our phosphotyrosine residues, right? Remember all the way up here, guys, we talked about the protein. Technically, this is an here. We'll say this is an amino acid AA, right? And then there's a bunch more AAs over here, and that whole thing makes a protein. So here's your amino acid. That amino acid is going to bind, instead of having the OH group, it's going to bind this phosphate over here. Yeah. And at that point, let's say that this is a tyrosine, and it binds the phosphate over here, phosphotyrosine. Okay, just to help again, nomenclature, very important. Where were we? Here. These are going to bind the SH2 domain. That just is talking about a specific part of an oncogene or a proto-oncogene. A specific part, a certain sequence of amino acids are called the SH2 domain, and they're going to bind phosphotyrosine residues. SH2 phosphotyrosine, SH2 phosphotyrosine, SH2 phosphotyrosine. Lock it into your head. That's important. Okay. What about SH3? Here, let's make some more room down here. SH3 is going to bind our proline-rich domains. So a domain is going to bind a domain. Yeah, it's just a place. It's just a spot. It's just a certain area. That's it. Absolutely. Yeah. So, okay, here it tells us now. So we know that SH2 binds our phosphotyrosine residues, like we just said. SH3 is going to bind our proline-rich domains. Like we said, I'm gonna ask you guys this in a few minutes, so keep that in mind. I hope everyone will answer that question, even if you're on the recording at home. Say it out loud. See if you were right or wrong, because that's important, right? So now we covered the structural features, nomenclature, of non-receptor tyrosine kinases based on the Sark as the prototype, SH1, SH2, SH3. SH1 equals kinase. SH2 equals phosphotyrosine residues. SH3 equals proline-rich domains. Know that. Become comfortable with it. Here again is just another illustration of what we're talking about in a different way. This is a little bit more consistent with what you're going to see in the field, right? So you're gonna see like, oh, yeah, like plextrin. We're gonna talk about it later in the future. Integrins, which are gonna be important, uh, in terms of binding certain aspects of the basal lamina to the epithelial tissue. Again, I'm just showing you guys, eventually you're going to get into this discussion. Integrins are just hemidesmosomes. If you don't know this stuff, you're never going to be able to explain the many complexities that come afterwards, right? uh Junctional um systems, right? Complexes, tight junctions, junction junctional occludens, junctional adherens, or zonular adherens, zonular occludens, desmosomes. These things are going to be super important, I promise you, especially when you get to histology, which is gonna be part of your med school, nursing school, PA school discussions. I guarantee it. Okay, but you have to understand all this first. This is just showing you an overview. Here's our able, right? Our kinase domain, aka we know this, the SH1. Here's our SH2, which is going to be binding what? No cheating. SH2 binds what? Phosphotyrosine, PY. Look at, all right. All right. Yeah, we got fancy out here. I like it. Yeah. Yeah. Good. I'm going to just throw in random little questions like that. SH3 domain. Here, another one. There's a little bit more space between ZAP70, with ZAP70 between the kinase and the SH2. Oh, wow. Sometimes there's two SH2 domains. Wow. Okay, cool. Oh, here's our Jack with that pseudo kinase domain. If you see the pseudo kinase domain, right? Think if you, okay, now I have one and now I have two. Oh, one's real, one's fake, two-faced. I'm a Gemini, so I take offense to that term. But hey, kinase is pseudo kinase, right? You have two, Janice. Oh yeah, Janice, that's right, the two-faced girl lady from Office. Oh yeah, he's the god of the two-faced god. I don't know if you watch Game of Thrones either. Listen, I'm a nerd. Let's get that out on the table now. The many-faced god, right? Just think the two-faced god is Janice. Okay. Jack, Janice, kinase, Jack. Great. Okay, we see that. We're talking Jack. Might be a question. Who knows? They're not going to ask you about this yet. Fact, no. Sark, yeah. This is the main one that we're talking about. CSK Fest. Yeah. All right, just to show you there's variation. There's variation. They don't all look the same. Some have two SH2s, some don't have any SH3, some have pseudokinases, some have plextrin integrins. There's different domains. All right, point made. Again, showing you the big picture over here, right? And this is actually showing you the numbers associated, right? Here's a myristylation site. We're going to talk about myristylation, and there's farnesylation and geranylation when we talk more. I'm not sure if you guys even covered that yet. That should have been a big part of cell membranes, but if not, it'll definitely come up in lipids and cholesterol synthesis and metabolism. Oh, you did. Oh, beautiful, beautiful, good. Yeah, so you know what I'm talking about. Myristylation, this is where it's going to attach. Yeah. Oh, SH3, proline rich region. Oh, SH2, phosphotyrosine, kinase domain, active loop, phosphorylation, which we're going to talk about. Inhibitory phosphotyrosine. Oh, okay. So there's activating and there's inhibitory ones. Okay. See, it gets a bit more complex. We'll get there. Oh, here's what we're looking at now. Let's orient ourselves before we jump into something. This looks like a whole bunch of like an abacus. This looks like this is confusing to me. What in the world are they showing me over here? Okay, let's look at what we have here. ERB, I don't know what that is. I don't know what that is. VEGF, no idea, no idea, no idea. Okay, so I don't know what any of these are. Okay. Wait a minute. This looks kind of like a cell membrane over here. Oh, it is a cell membrane. All right, yeah. So that means it already tells us in the title, we're looking at protein tyrosine kinase receptors, which we know are single transmembrane spanning proteins. I showed you that in the first picture, right? When we drew the cell membrane, we had the outside portion, the ligand binding portion, the transmembrane domain, and then the catalytic domain down here, right? We talked about that. Oh, okay. Oh, look at these things. These are little domains like we saw. Oh, these look like little kinase domain, little SH1s over here, these things. Okay, so if these are on the inside, and they are on the inside, we know that because they're kinases, right? We're assuming that these are all kinases, and because here we have all these little additional pieces, then this must be the outside. So yeah, this is in the cytoplasm. Oh no, my bad, my bad. I gotta switch up. I gotta switch up. Yeah, let's not even get into, yeah, just disregard that, guys, because I think I messed that up. Yeah, so these are the receptors. My bad, my bad. These are the receptors here. I was trying to get a little too cute over there. My bad. All right. So here, let's just break into it. List of protein tyrosine kinase receptors. And you might get an image like this. That's why I was trying to kind of break it down, uh, to make it a little bit clearer. But these are the main ones you have to know. Okay, so this I got it switched up. Let me get rid of all this. Okay. ERBs, EGFR, HER1, HER2, insulin receptor. There's a few of them. IGF-1R. I don't think we're really going to get into any of these too much, but we are going to get into these. And this is kind of just showing you there's quite a few different ones. We're only going to go over a few of them. Okay, I'm going to definitely clean this up for the other portion. I might not even mention this because I think I just confused everyone. Disregard all that, guys. My fault. See, this is why Dr. Larea is such a genius at what he does, because he can take things like that, and he explained this, and he can make it sound like, oh, I get it. Then I go to teach, I'm like, what? All right, cool. Receptor tyrosine kinases. Okay, so here's the general outline. Okay, we're gonna have step 1, step 2, step 3, step 4, step 5, step 6, and they're all gonna link to each other. Now he's just showing you step 1 to step 6, kind of, right? Arbitrary numbers. Don't pay attention to that. But outside of the cell, here, at least it's labeled here, outside of the cell, inside of the cell, membrane. Okay, so here's our transmembrane domain. Here's our ligand binding site on the outside. That means this must be the receptor portion over here. Extracellular ligand binding domain, transmembrane domain, intracellular portion containing the kinase domain. Remember, the intracellular portion is where the catalytic activity is going to be. And of course, a kinase is catalytic activity. It's catalyzing a reaction. It's adding a phosphoryl group. It's phosphorylating. Yeah. Okay. So session objective highlighted. Our eyes should be directed here now. Session objective: explain how signal binding to catalytic receptors leads to the activation of signaling pathways and biological responses. That is the session objective. That is, in other words, the point of this whole lecture. Great. So we're going to pay attention to that. That's the question we're going to keep in our head now, because we covered a lot. Now we know the general basis of what we're talking about. Uh-oh. Let's not get into that yet. We don't want to break any hearts just yet, but just remember, ligand binds to the extracellular side. Intracellular side is going to evoke the reaction. Okay. Keep that in mind. I'm going to ask that again later. Who can tell me what the SH3 domain does? First one wins. Yeah, Tanishka, let's get it. All right, yes. Very nice. Yeah, proline rich. I'm gonna throw in a few random ones. Okay. Whoo, that is a lot. We are looking at a lot over here, folks. Okay. Let's just try to get oriented really quick, because I'm getting dizzy just looking at this over here. Okay, so we're looking. What do we recognize? Oh, RTK. First of all, RTK is what? RTK equals tyrosine kinase receptors. Don't ask me why it is that way. It is RTK. If you're talking about RTK, we're talking tyrosine kinase receptors, which means what? Oh, okay. Yeah, we talked about this. Tyrosine kinase receptors. They have the extracellular ligand binding domain. Oh, okay. So these must be the extracellular ligand binding domains over here. Okay, that makes sense. So that means this is extracellular. This is cytoplasm. Okay, good. This is obviously a cell membrane. We can tell that pretty easily, right? Oh, okay. So if we have the extracellular, now we know this is all intracellular inside the cell. And that is where our catalytic activity is going to be happening. Okay, good, 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 great. Beautiful. Now we got to deal with all this fun stuff. We're not going to make you learn all of these, or rather, the course faculty will not make you learn all these. I don't really have a say in it, but you're not going to have to learn all of this in depth. Um, Granted, this is only a part of the whole story. All right, so I should rather correct myself. You don't have to know the entire story. This is only showing you a small little section of it. But a good amount. Like I know for a fact you're gonna have to know the RAS pathway. That's for sure. We're gonna go into PI3K and P10, of course. Jack stat. Here, see Jack stat. All right, so you're gonna have to know a good amount of this, but consider yourselves lucky because this is a much bigger diagram if you include all of the stuff. Okay, I have to double check with Dr. Larea on exactly which exact mechanisms he wants me to go through. But yeah, so welcome to the big leagues, folks. This is going to be a good time, and we can make it fun. Okay, let's talk about this pathway here. These are rather pathways, I should say. There are quite a few. Okay, so here we have. Notice that there are two of them. We're going to talk about what's called dimerization, and dimerization is often a big pre-step in terms of what we have to be doing. Okay, this is a big one. This is one that we're going to discuss in great depth. And we talked a little bit about JAK stat. We know what JAK stands for, right? The Janus kinase. Because it has what? It has a kinase and it has also a pseudokinase domain. Oh, P10. We saw that in one of the questions earlier. Oh, wait a minute. This has a little line here. This usually means inhibition. This usually means it stops it. Okay, so maybe P10 stops PI3K, huh? I don't know. We're gonna have to go on and find out. What's this? Hematopoiesis. Oh, hematopoiesis, the creation of heme. Or is it blood? We're gonna find out, guys. We're gonna talk all about this. Oh, and remember these big words that we talked about? These important words: metabolism, differentiation, proliferation, and survival, slash apoptosis. Wow. Okay. I noticed that all three of these pathways are pointing to this big red box. So I'm trying to tie in what we talked about initially. I'm trying to tie in, okay, we have a receptor, we have an intramembrane domain or transmembrane domain, we have a catalytic domain over here, and then after that, we got a bunch of steps. We got a bunch of steps. We got a bunch of steps. All those bunch of steps, which, yeah, you're going to have to know them. Sorry. They are ultimately trying to do this. How much is it going to grow? What's the cells going to turn into? And will it die or will it live? Okay, that is what we're trying to focus on here. Besides the JAK stat, which goes into the hematopoiesis aspect of things, and that doesn't really have that's really just making blood at that point. So, okay, the beginning. That's what's crazy is we just hit the beginning, right? We're almost 30 slides in, 25 slides in, and we just hit the beginning. And I feel like we already learned a lot. Yeah, we broke it down pretty into little small pieces, but this is just the beginning, unfortunately. Um, so let's just jump right in. Okay. I only have about 15 more minutes until 4. Okay, which is the main reason I have to break these up into other pre-recorded lectures that are premeditated, so I'm not just rambling on as much as I did today. Just make some more concise statements. But yeah, so we're gonna just try to get these little beginning concepts finished up. Okay, and then after that, guys, it really is just a matter of memorization. Okay, so if we're talking about the RAS map K pathway, like there's gonna be map here, let's look at it here. RAS map kinase, map kinase, and then and it just goes on and on and on. Like it's gonna be memorization. Me explaining it won't help much, so they're not gonna be super long videos to watch. I know you guys don't need more stuff to watch, and again, these are optional, but this is a brand new subject for most people. It was for me at least. And I took biochem in undergrad. I killed it in undergrad. And when I saw this, I was like, what in the world subject? What class is this anymore? But yeah, so we're gonna do what we can here, guys, and I'll let you go and you know, regain your sanity a bit, and then I'll torch you a bit more on those pre-recorded videos later. Okay. Concept. Ooh, this must be important. It's bold. And it's in a little blue box and a red box that I made for you guys. In the absence of agonists, the receptor is in the off state. Oh, that we said that already. Okay, so that means this is off. Oh, these must be on, right? Because there's receptors there. But what's different that you notice between this one and these ones? Well, I just said it. I kind of just said it. I kind of told you. These ones, there are two now. Here's only one. Here, there's 1 and 2. Dimerization. Dimerization. Two. Dimer. Something a two component thing, a dimer, right? If I have a homodimer, homo from the Greek same, I have two of the same thing coming together and forming a two component unit of the same components, right? So if I have uh, A and A, whatever it may be, right here, you can say it's an apple, two apples, and I want to make a homodimer. A. Now they're just together. They're linked up somehow. That's a homodimer. Now, if I have A and here, O, an orange, a little green leaf over here. Now we're comparing apples and oranges, right? These are two different things, but we can still put them together, right? They're still fruit. Have you ever seen My Big Fat Greek Wedding? It's a funny movie, right? They're still fruit, but now we're talking a heterodimer. Hetero, different. Homodimer, the same. Okay. You'd be surprised how few people actually are not familiar with the term homodimer or heterodimer, and again, I wasn't, but sometimes these uh very intelligent professors they live and breathe this stuff and it's second nature to them. But for us lowly grad students, I'm like, what in the world are you talking about? Yes, the activated one is a dimer, exactly right. So usually when we see activation, at least when it comes to tyrosine kinase receptors, if we see activation, we usually also see a consequent homodimerization. Okay. Now, what's the point? Why do we need these two to come together? Why? Why? It just sounds like extra work. Well, there's a reason for it. And that all comes down to this lovely concept called autophosphorylation. Auto, auto. We know what phosphorylation is, right? I'm not going to go through that. Yeah, you're phosphorylating. Kinase phosphorylates. Kinase action, right? We're talking about tyrosine kinases, right? You're adding a phosphate. Okay, we know that. Auto. What does auto mean? Self. Self. Automatic. Yeah, something is doing it itself. You don't have to do it for it, right? If something is automatic, the car is automatic, it's shifting gears by itself. If it's manual, you got to use the stick, and the whole thing. So auto means self equals self. Yeah, I'm gonna break it down into these little tiny terms for you guys. All right, because if you guys got it totally from Dr. Lagreya, I applaud you. And then you don't really need this review session. But I'm talking to those of us who were kind of more like I felt when we were in biochem, and a lot of these words were foreign to me. So auto phosphorylation, self phosphorylation. Oh, they're phosphorylating themselves. Okay, okay, but that doesn't make sense. If there was alone, they can't phosphorylate themselves. That doesn't make sense. Oh, they wait a minute. They phosphorylate each other. Oh, okay. So this isn't really autophosphorylation that we're looking at over here. This isn't really autophosphor. This is trans autophosphorylation. What does trans mean? Trans. We broke down auto. We broke down phosphorylation. We know what this means. Or phosphorylating itself. Trans means what? Trans means yeah. Sorry. Exactly. Between trans, the other side. Yeah. So other between I don't know the exact word, but yeah, it's if you're trans autophosphorylating and you have a dimer situation, right? So you have the same exact thing coming together into this, right? And you want to trans autophosphorylate. This one is going to phosphorylate this one, and this one is going to phosphorylate this one. It's bro. Okay. I think it's pretty straightforward. I'm not going to belabor the point. Okay. But you guys get it. Long story short, one, two. Okay. Ligand comes in, binds. That binding causes what? Causes these two to come together and become this, a dimer. What kind of dimer? A homodimer, because they're the two exact same thing. They have the same exact structure, a homodimer. Okay, so now we have the homodimer, and once we have that homodimer, there's a an interaction that occurs between the two, right? So here, let me erase a little bit. We're gonna imagine that here, because usually we have the SH1, which is what? What's the SH1 domain? Kinase. SH1, which we never really use here. Kinase. Usually that comes first, right? SH1. Yeah, makes sense. Okay, so we're going to assume that these kinases here, if they're participating in this trans autophosphorylation, we're assuming that this kinase is going to throw a kinase onto here, and that this kinase is going to throw a kinase onto here. Oh, what do you know? Yeah, that's exactly what we got. Look at that. So let's assume, I'm going to ask you guys a question. Let's assume, I hate when it does that. Let's assume, we don't know for sure, but let's assume that this is SH2. These pink domains are SH2. What is this that the phosphate is sitting on? Which one is it? P or Y? Or PY? Tyrosine. This is the Y. Because this is just nothing but a residue, right? These are all just, this is an amino acid chain, right? And they don't, of course, look nice and linear and straight like this. They're all contorted and combobulated into secondary, tertiary, quaternary. Well, yeah, only tertiary structures, because this whole thing would be a quaternary structure, the dimer itself, because there's two different protein chains. But this is just one long chain, and they make it look pretty like this. But yeah, there's a tyrosine in here, and of course, it wouldn't just be one tyrosine in such a large chain. There would be little tyrosines everywhere. They'd be all over the place. But for simplicity purposes, they put the one phosphate onto one spot. If this is SH2, yeah, that's a tyrosine. That's a tyrosine. What if it was SH3? Then what are we looking at? A proline rich region. Yeah. We're going to see a bunch of prolines right next to each other, and that's where the phosphate is going to be added. That's where the kinase is gonna direct its phosphorylation. All right, that is important. Be familiar. Each monomer is both an enzyme and a substrate and an allosteric activator. Right? It's an allosteric because it's not going to the active site, but it's binding with the other portion of the dimer, right? The other monomer, and that binding is causing activation of the kinases, and the kinases are binding or are adding phosphates or phosphoryl groups, whatever you want to call them, onto wherever it's told to bind. Right? If it's an SH2 domain, we're talking the tyrosine. If it's an SH3, we're talking proline rich domains. So these are important. This I would make a test question. I'm not saying our instructors will. I might do it. Why? Because that's pretty cool. And if they were to ask you that question, it would be totally fair. But you know what? You might be too busy learning. Oh, here the JAK-STAT is this pathway, this path. This might be important too. And I hate to be that guy that's like, everything on every slide matters, because you know what? That's not always the case. But in this particular lecture, if it's on the slide, know it. Okay. There's certain things that are highlighted, certain things that are uh um boxed in or whatever. Dr. Larea does everything he does is with a purpose, or at least so I've learned. I'm confident the other instructors know exactly what they're doing as well. I just don't have as much experience with them as I do with Dr. Larea or Dr. Peterson, because they are either my current or my previous instructors. And Dr. Larea did do coag with us, but we didn't have cell signaling with Dr. Larea when we were taught. We had Dr. Olson, and yeah, so I've never actually heard Dr. Larea teach cell signaling before, but you know, if there's anyone who should be doing it in place of Dr. Olson, it's him, because he's also huge with the cell biology um, department as well, and that this is a huge aspect of cell bio. I think everyone has to do cell bio in their final semester, and that's supposedly the hardest class at New York Medical College. So you learn this, you're gonna do yourself a big favor and make life a lot easier. Okay, so mechanisms leading to unregulated. Now we have a problem. Now we're talking issues. I'm not going to go on much longer, guys. Just bear with me. Okay, we're almost done, and then we'll get into the memorization parts later. It's where you can just keep on going back and back and back. But I think you guys are all up to stuff over here. You're good to go. Unregulated, bad, right? Skull and crossbones. We don't like that. Okay, so now we're going to see overexpression of RTK, tyrosine kinase receptors. If we have too many receptors, right? Too many of the first step, and we just talked about amplification, right? Even this is normal, right? This is how it's supposed to be. Let's assume that the rest of this is filled in. One receptor gives me 1,000 end products, whatever they may be. If I have an overexpression, what does overexpression mean? You'll keep on hearing that. It means the genes have been coded to build more of these tyrosine kinase receptors. It's being expressed more. We're seeing more of them, right? Is a greater expression. Overexpression is too much. There's too many present. There's too many receptors present. What does that mean? Even one more receptor brings us from 1,000 to 2,000. Does that sound like a problem to you? One more than we're supposed to have can double the problem. Of course, we would not just ever have one receptor being simulated, but receptor theory is a whole different concept we'll get into when in future courses. But yeah, that's a problem. Yeah, so over amplification. Okay, now we have way too many signals being thrown out down here at the bottom part. Okay, if you have too many signals going out, you have what? What are these signals' ultimate job? What are we focus? What's the final product, the whole goal of all this stuff? Yes, metabolism, differentiation, proliferation, and apoptosis, or cell survival, right? Those four things: metabolism, proliferation, differentiation, or apoptosis. Okay, so if those things aren't working right, we have hyper expression, overexpression, over amplification, too many end products, which means we have too much proliferation, too much gene transcription, too much differentiation, too little apoptosis, or too much. Who knows? Maybe we have too little of all of these. Not a right amount. What does that mean? Greater chance for mutation. You have more cells being produced than you need to have. Law of statistics, guys. Something is bound to go wrong eventually. If we live long enough, if the human body was able to somehow survive to 200 years, the chances of you getting a cancer are extremely high. Why? Because you keep on dividing and dividing and dividing. Imagine like you can be the best worker in the world, right? You can let's say you're learning your amino acids, right? You guys were studying for your exam. You're learning your amino acids and you got it down, right? You've done it 1,000 billion times before. I wrote out the sheet a million times. I wrote it out. I know my amino acids. Okay. You think if you did that 300,000 times in a row for years on end, do you think at some point you might make a mistake? You might not draw in the negative sign on one of the oxygens. You might accidentally put too few carbons. Yeah, that's what's happening here. You're putting too much. You're making the statistics like more likely to happen. That's why he says mutation here. Overexpression of RTK, too many receptors, too many end products, too much proliferation, too many cells being made. Too much labor means higher chance of error. One error in the right place, one mutation leading to either truncation or other anomalies. Truncation meaning it just diverges from the typical pathway. Just one point. A point mutation is one single amino acid residue in a line of thousands, right? That one single, what we talked about, that one single valine to phenylalanine can be a big problem. And we saw that because it caused some type of oncogene to form from a proto-oncogene. 1 point mutation in the intracellular domain, intracellular, why does that matter? Because that's the catalytic part, can result in gain in function. Okay. There's also autocrine loops that you can discuss. We'll talk more about that as well. That's basically the receptors themselves shoot out ligand that so they're going to make their own little ligand, and that's going to keep on activating itself, and it's an autocrine loop. Autocrine, it automatically sends like a crine signal. I guess there's paracrine, endocrine, the autocrine. But yeah, this is pretty much activating itself. So these are just some of the ways that, an unregulated mechanism can cause a problem, right? And here, this is the first step, right? So this is the first step. I'm gonna stop here, okay, because now we're actually talking RAS, RAF, MEC, ERK. You know, these are the actual, GRB2, SOS complexes, and we're not going to go over time, but I'm going to break these down piece by piece, not as as intricately as I did for the first half of this lecture, but it really is important that I pointed these things out for a reason. So pay attention to the things that I highlighted. You see, I already went ahead and kind of gave myself a few notes to go through, but we're gonna stop here. Are there any questions you guys have just for now? I mean, if you do have them later or something, if you want to look over this again and think a little bit about it first and then come back to me, that's honestly probably better because then I can include it in the recording for everyone to see. Oh, when it says monomer, is it talking about the tyrosine residues or is it a domain? So a monomer means one piece. It could be talking, and no, it's not talking about the residues. A monomer is this. This is the monomer. That's the one piece. This is a monomer. One piece. It's one independent entity. Oh, that was for the previous slide with autophosphorylation. Let me take a look. Yeah, same thing. You're talking about this part here, right? Yeah. So each monomer is both an enzyme, a substrate, and an allosteric activator. So let me just run through that really quick. Yeah, that's still talking about these individual pieces, right? Because remember in enzymology, a substrate binds to an enzyme to give you the enzyme substrate complex, and then that gives you the reaction, right? And the enzyme, we're basically calling this the enzyme, right? This, let me get a different color. This is the enzyme. This monomer, this is one piece of a dimer, right? The whole thing is the dimer. This one piece is the monomer. Yeah, so this functions as an enzyme. In other words, this is what will get activated, right? This is going to have a substrate come in and bind to it, thus activating the enzyme, right? Once an enzyme is only active when there's a substrate bound to it. That's like the first rule of enzymes, right? So this is more like theoretical. This is kind of just like to help tie in past information, right? Of course, it's not an actual enzyme, but it is an enzyme in that it takes on a substrate and that the binding of the enzyme with the substrate will cause the reaction, right? Because once this happens, we have the reaction. Okay, so if this is the enzyme, what's the substrate? This one. The other one is the substrate, right? And vice versa. So this is also an enzyme, and this is also a substrate. That's what he means here. Enzyme and a substrate. So they're both, because this one is acting as the substrate for this one. This one's acting as the substrate for this one. And this one is acting as the substrate for this one, right? Trans autophosphorylation in a dimer of non-active kinases. Now, in order to get activated and to give off that signal, we have to phosphorylate, right? And that's kind of the whole point here. They have to dimerize before they can phosphorylate each other. Makes more sense? It's totally cool, guys. That is a crazy, like, it's a totally like strange concept. All right, cool, great, yeah, awesome, yeah. What else? What else can I clear up now? I really appreciate you guys being so active and for bearing with me earlier. I for having to switch things up. Um, yeah, I really appreciate it. You guys are definitely super active, and it makes these sessions a lot easier. Yeah. All right, cool. So if there's nothing else, I'm gonna go ahead and wrap this up. Just as a reminder for anyone watching the recording, I hope you're not cramming right now. You know, if you if you're watching this a few weeks later, um, definitely do your best to focus on some of the nuances and the subtleties, because as you saw in the first exam, a lot of the questions are not just what is blank. It's they present you with a situation and you have to tie in a whole bunch of different, concepts to come up with the right answer. All right. Um, I'll put out those recordings either tonight or tomorrow morning. Um, Depending on how things go. I just got to reach out to Dr. Larea and just get a little clarification on exactly what he wants done. If anyone has the second half of that recording, that would literally be awesome. That would be life changing. If we had Dr. Larea last year, I could have just used my own recordings, but we had Dr. Olson, so yeah. And then blood coag is it's not terrible, so I'll run through that as well, pretty quickly. But he really does it. He does it. There's no way I can explain it better than Dr. Larea does. And there you have the full audio and full video. So I'll certainly talk about it and answer questions, but your best bet is just watching Dr. Larea's video, because you know he makes it pretty straightforward. All righty, y'all. I will see you at the next one. Email me with questions. Definitely email me. Hit me up. Have a great one, guys. You're welcome. Thank you. You guys are the best. Have a good one. All right, all right. No problem, guys. For sure, for sure. All right. There we go. End meeting for all.